Alright, so today I'm going to be talking about CDK and cyclins. Unfortunately, I can't be with you in person. I was exposed to COVID last week, and we'll be doing this presentation from quarantine, but we're going to make a video and make this work. So first, talking about what cyclins are. They are proteins that are going to activate CDKs to advance certain events throughout the cell cycle. CDKs then are cyclin-dependent kinases, so they're the actual kinases that are causing phosphorylation reactions to occur to advance past certain checkpoints and stages in the cell cycle. So cyclin-dependent kinases require the binding of a cyclin in order to do their job and to be fully active. With this said, cyclin concentration throughout the cell cycle is somewhat volatile, depending on what cyclin it is and what stage of the cell cycle. Different cyclins rise and fall in concentration pretty variably. CDK, on the other hand, will remain fairly constant and consistent, but getting more into the mechanisms of how this cycle works and what the biology is behind it. CDKs are indirectly regulated by p53, which activates cell cycle arrest in the event of DNA damage. So when G1 is initiated, cyclin D begins to drive G1 and the events within G1. And so CDK4 and CDK6 bind to cyclin D, and binding begins to inactivate RB, the retinoblastoma tumor suppressor protein. And RB loosens its grip on E2F, and then a new CDKcyclin complex, cyclin E CDK2, comes in, and that further phosphorylates RB to inactivate it and fully release E2F. So this allows the cell cycle to progress from G1 sort of in that transition towards the S phase. And cyclin E CDK2 also phosphorylates P27-KIP1, which is an inhibitor of cyclin D that degrades itself. So CDK2 along with cyclin E promotes this transition from G1 to S, and this degradation of P27 along with E2F release promotes production of cyclin A. Cyclin A binds to CDK2, and that fully promotes entry into the S phase. So E2F removes a cell cycle response element to begin transcription of cyclin A, and so there's a negative feedback loop here where cyclin A phosphorylates E2F to prevent it from overproducing too much of cyclin A. So there's some checks and balances in play there, and that's important to keep the cell regulated. And if one of these functions ceases, that's one of many ways that cancer could be induced. All right, so moving on, cyclin A now wants to promote mitosis in G2, so it binds to CDK1 now rather than CDK2, and this is going to promote the entry into mitosis. However, we need to create cyclin B, CDK1, so as a result, cyclin B begins to be produced, and it takes the place of cyclin A CDK1, which helps stabilize it and is then degraded. Cyclin B CDK1 is also referred to as maturation or mitosis promoting factor, also called MPF, so I might refer to that throughout this rest of the PowerPoint. Okay, so here's, again, there's so many different functions of CDK and cyclins, and I see that I'm already past 4 minutes, so I don't want to spend a ton of time here. But cyclins are huge in all kinds of processes within the cell cycle, maintaining the fidelity of DNA sequences, even in some regulatory and hormonal endocrine functions here with glycogen and insulin secretion. So a variety of functions that it has. And Dr. Blumenthal wanted us to draw our own little cartoons, so here's my cartoon, or one of them. And just want to point out, I don't know if Zach is in the class today, but this was created using G2s, and he's a big fan of G2s, so throw that out there for him. So again, this is not necessarily drawn to scale because mitosis is going to typically be a very small sliver of this whole cell cycle. However, just for the purposes of illustrating it, I kind of fanned that out to give it more space. So we start here at the top and we're moving clockwise. G1 has the option to go into a state of G0 and no longer grow. It needs the correct growth factors and mitogens to move this process along. It also needs cyclin D and CDK4 or 6. And there's 3 different checkpoints here. The one from G1 to S, which is regulated primarily by cyclin E CDK2. There's one later on here regulated by MPF, which is between G2 and mitosis. And then there's a spindle checkpoint between metaphase and anaphase within the mitosis phase. So at these different checkpoints, there's different questions sort of that the cell asks itself. If DNA is damaged, P53 can help that. And it just has to check all the boxes, and that will help it move on to the next cycle. Again, I talked about the sort of volatility, the rise and fall of different cyclins. In class, we learned that cyclin D is pretty much constitutively expressed or active, and while that's not entirely true, it certainly is more stable relative to the other ones, which are going to rise and fall a lot more abruptly. And this is my second cartoon that shows just how complicated the relationship between cyclin and CDK is. There's definitely some checks in play to make sure that cancer doesn't happen. And so you see here, CDK and cyclin bind to each other, and then we've got some phosphorylations that occur. CAK, that's cyclin activating kinase, adds one phosphate to CDK, and this WE1 is going to add a second one, and this second one is actually inhibitory, so it helps cyclin bind a little bit better, but it prevents full binding. And so for that, we need CDC25 to dephosphorylate to fully activate the CDK-cyclin complex. And as you can see, it becomes fully active here in the far right. So cyclin CDK cancers are all over the place. In 50% of breast cancers, cyclin D is overexpressed, which is a huge number there. Cyclin B found to be implicated in colorectal, lung, and squamous cell carcinomas. Cyclin E in lung, gastrointestinal, breast, lymphoma, and leukemia. And then I want to take a second here to look at this next table. Hopefully you can see it, but there's this whole table of how each of the CDKs here are involved in all kinds of cancer, from melanoma, glioblastoma, adenocarcinoma, all kinds of pancreatic cancer, breast cancer, colorectal, just a huge variety really that CDKs have an influence in. And it's not surprising really to see that play out, because there is just so much happening within the cell cycle that if something happens that's not good, even at one point, it can be disastrous for the cell cycle and result in cancer. Here we're going to look at 2 drugs, flavopiridol and dinuciclib. No clue if I said that right. But this table shows that they've been tested for a variety of cancers in combination with a variety of drugs, and this helped inform the primary research article that I found, which I'm getting into next here. So Zhang et al. 2018, they explored CDKN2A, and CDKN2A is an inhibitor of CDK4 and 6, and specifically CDKN2A encodes for P16 INKA and P14 ARF. So I'm just going to call them P16 and P14 for simplicity's sake, because that's a mouthful. P16 functions in the inhibition of CDK4 and 6 to phosphorylate RB and block the G1 to S transition, whereas P14 activates P53, and P53 can then do its DNA repair functions, cell cycle arrests between G1 and S, induction of apoptosis, and even the senescence of short telomeres. So when CDKN2A is not being expressed, there's huge issues because you don't have any more P16, and without that, RB is constitutively phosphorylated to advance the cell cycle, and that's one way that it can cause cancer. And without P14, P53 is now inactive and unable to carry out its protective functions within the cell. So with that being said, CDKN2A is extremely important, and it's been found already to be inactivated in 72% of squamous cell lung cancer. So this research study looked specifically at squamous cell lung cancer cell lines and asked the question, do flavopuridol and dinucyclib, these two anti-cancer drugs, do they truly exhibit anti-cancer activity? And so they did this in a few different ways. First, they looked at cytotoxicity through cell viability assays to see if these drugs induced cytotoxicity to kill the cells. And then they also looked at, do these drugs induce apoptosis? So looking a little bit more mechanistically, do they induce apoptosis? And they use these Tunel assays, no clue how to pronounce that, and Western blot analysis to determine this. And so the 4 cell lines that they used in the process were H520, SKMES1, H1703, and H596. H596, has the wild type CDKN2A, which means that it's supposedly going to be keeping things in check more so than the other cell lines. All right, so the results of this, first of all, for the cell viability assay, found that there was increased cytotoxicity in the CDKN2A deficient cells. Or I shouldn't say deficient because one of them was just an inactive version of CDKN2A. But regardless, cytotoxicity was induced, cell viability was lower. So as you can see, the H596, which has the wild type gene, it had the greatest cell viability, whereas the deficient or inactive versions of the gene had much lower, especially as concentration of the drugs increased. And this is good because that means that the drug is resulting in cancer cells not being able to grow if they're deficient in this factor, in this gene. Over on the right, we've got flavopuridol inducing apoptosis in the cancer cell lines that are inactive or deficient for CDKN2A. And the same with the dinucyclib, and the Western blot just further shows that, as I'm sort of running out of time here. So overall, these results show that both cytotoxicity and apoptosis are induced through these drugs, and they are able to specifically target the cells that are either deficient or having an inactive version of CDKN2A, which is good. So here are the sources that I used to create this presentation, and I can't really field your questions, but I suppose if you want to email you can. But yeah, I hope you enjoyed my presentation, my cartoons, my research article, and enjoy the rest of your day.